Good morning, everyone. New chapter today, chapter 3, energy release and the breakdown of organic molecules. Number 1, basic recap on cell respiration and fermentation. So basically, we are going to follow the same process as for photosynthesis. We're going to study the gas exchanges. Then we'll have a look at the organelles, and then we'll have a look at a bit more detail, which will, just like in photosynthesis, involve proteins, ATPA synthase, and oxidoreduction reactions. So in many ways quite similar in many aspects as to photosynthesis. 1.1, cell respiration. So we're going to sketch the experiment, or a experiment, which could prove the existence of cellular respiration. So we're going to need this kind of apparatus if we were to make a diagram of it. This is just a diagram of the apparatus, and on the next slide we'll do the diagram you actually need to do. So these kinds of apparatuses have two probes, O2 probe, CO2 probe. Then we have a cover plate. Then we have a bioreactor tank. That's where we can put the yeasts. Shutters, that's in the case of if we want to try without or with light. That's more for photosynthesis really. And a mixing speed knob just to make everything mixed up. So this is the sketch you can do in your booklets. So we're going to add two probes. So this being the bioreactor. Obviously in this bioreactor, in the water, there is a certain concentration of dioxygen, a certain concentration of carbon dioxide. And in the middle of this we're going to have a few yeast. Obviously these are, you've probably guessed, they're not exactly what yeast look like. We'll have a look at a microscope picture just afterwards. What's important is that if yeast take in O2, that means that in the medium there'll be less dioxygen, and we will see on the graph that the dioxygen concentration decreases. For example, if CO2 is produced, if there is more CO2 in the medium, which would be measured by the CO2 probe, that would mean that yeast release CO2 into the medium. That's the basic concept of these bioreactors and the use of probes. This is a microscope picture of yeast. It's obtained with a light microscope, just like the ones you can use in class. You need an idea of the scale. I don't know if you have any idea how big a yeast is. Well, here's the answer. It's roughly 4 micrometers. You can nicely see the nucleus here, or there. So it's one cell big, and they do, that's probably what's happening here, they do simple division. You probably have just, no sorry, these two, you just have the end, they're splitting here, so it's simple division just like for bacteria. There we are, that's what yeast look like. Well, now we're going to analyze the results that have been obtained with this experiment. So you've got the graph. We're going to pinpoint the important parts of this graph. So first of all, we're measuring two gases. So the O2 concentration, the unit is in milligrams per liter. Then we have also the CO2 concentration in milligrams. So the arrows, obviously, they're pointing to each y-axis. So the blue line is CO2 and the O2 line is dioxygen. Now then, we need to analyze the concentrations. Oh, you might be wondering why there's this line here. This is to state that glucose has been added. Glucose. So with a simple syringe, you can add glucose. There are usually holes in the bioreactor for that. So at 300 seconds, the x-axis being time in seconds, glucose is added. And we might as well add the titles, variations of O2 and CO2 concentrations in a medium containing yeast. So we're going to split this into two, before glucose is added and after glucose is added. It seems to just slightly increase, but that could also be, it's probably not significant compared to the precision of the probes. So we can say that for the CO2 concentration that we're reading on this y-axis here, it remains constant at 6.6 milligrams per litre. Don't worry if I'm going too quickly. On the next slide, you will have all the text with the analysis. So I'm just going to say it quickly and then we'll go to the next slide where you can have time to write it down. So it remains constant at 6.6 milligrams and then it increases from 6.6 all the way up to 10. That's for carbon dioxide. What about oxygen? Same thing, it might slightly decrease, but we're going to consider that it remains constant at 11 milligrams per litre. And once the glucose is added, we can see that it decreases from 11 milligrams per litre to 1 milligrams per litre. That's all for the analysis on the graph. I'm going to move to the next slide so that you can have the detailed elements. So as we did it, we split the analysis in two, before glucose was added and after glucose was added. Before glucose was added, we can state the following. It remains constant at 11 milligrams per litre and the CO2 concentration remains constant at 6.6 milligrams per litre. So I invite you to pause the video and write this down. After glucose is added, we see two things: a decrease of O2 by 9 milligrams and an increase of the CO2 concentration by 3.4 milligrams per litre in 800 seconds each time. Well then, what can we conclude? We can conclude the following. Very basically, in the presence of glucose, yeast take in dioxygen and release carbon dioxide. That's the more global conclusion, and we can perhaps assume that the O2 that's been taken in, perhaps it's consumed and the CO2 is probably produced. But strictly speaking, with this experiment, we could only state the first one. That's it for this first part on respiration, and we're going to do fermentation now. 1.2, fermentation. The first question we're going to ask ourselves is, what's going to be the difference between the previous setup and this one? The first setup was for studying respiration, and this one is for studying fermentation. Well, in fermentation, one of the key things is that there is no dioxygen. Therefore, the setup must be air-sealed. The bioreactor where the yeast will be placed must be air-sealed so that no dioxygen can come inside. We're going to do the same thing. We're going to analyze the graph. So similar setup with different probes, 3 probes this time. as to measure dioxygen, carbon dioxide and ethanol. The formula of ethanol being C2H5OH. All these concentrations are in milligrams per litre. The blue line corresponds to dioxygen, the green one to carbon dioxide. Okay, we're going to split the graph in two. There's something interesting that happens at 200 seconds. The amount of dioxygen is down to 0. There is no more dioxygen left. And you can see that more or less it corresponds to the moment where ethanol starts increasing. So basically what's happening here is that in this first part here, there is still dioxygen. So in this part, the yeasts are doing respiration. Respiration in this part. And in the next part, they will be doing fermentation, in which case ethanol will be produced. In both cases, carbon dioxide is produced, but the striking thing is that when there's no more dioxygen, then ethanol is starting to be being produced. Last thing I forgot to say is that at the beginning here, at the very beginning, glucose is added. So here we are. So just like last time, the detailed analysis is going to come up. Oh, I forgot about this. Time in seconds, and also variations of O2, CO2 and ethanol concentrations in a medium containing yeast. That's for the title. Analysis, so two parts, when there is still dioxygen up to 200 seconds and when there is no more dioxygen. So as we said, the CO2 concentration increases, the ethanol concentration remains constant at 0, so there's no ethanol when there is still dioxygen. And when the oxygen has disappeared, the CO2 will still continue to increase, but now there's a new element, ethanol also increases. General conclusion: when there is glucose but no dioxygen, yeast released and probably produce CO2 and ethanol. There we are, that's what you can write for the conclusion. So differences: respiration is you take in CO2, you take in dioxygen and you release CO2. And fermentation, dioxygen is not involved, but the yeast are going to release CO2 and ethanol. So that's the basics for fermentation and respiration, and we'll see more in later lessons. Just like for the plant cells, we are going to play the How Many Labels I Know game. You need to know the name of different compartments and organelles. No worries, because you already know quite a lot of them because they're similar to what you can find in the plant cells. Let's go. Number 1, the nuclear envelope, what surrounds the nucleus. Number 2, nuclear pore. They're the holes, if I may say so, which connect from the nucleus to the cytoplasm. Nucleolus, it's this well, not in the center, but this part of the nucleus where ribosomes are produced, for example. Number 4, euchromatin. So it's the chromatin which is with a lighter color. This means that DNA is less compact, and this means that DNA can be more easily transcribed into RNA in these parts, as opposed to heterochromatin. Heterochromatin is the darker parts. This means DNA is more compact, and therefore DNA is not as easily read to transcribe into RNA. Cytosol, so this is everywhere really. This is the liquid water-based solution of the cell. Then we have the plasma membrane, so you know quite a lot now about the plasma membrane. Mitochondrion, this is going to be the key organelle, one of the key organelles in this part on respiration. So we've got one here, got one there. Then we have rough endoplasmic reticulum. So this is this part here, rough because there are ribosomes on it, as opposed to the smooth endoplasmic reticulum, which does not have ribosomes. Bear in mind that the endoplasmic reticulum, proteins translate by these parts, and they can be modified and things can be added. Golgi apparatus, so they will concentrate different proteins, and same thing, things can be added, and usually vesicles leave the Golgi, and if cells secrete different substances, it will be through these vesicles that leave the Golgi. Lysosomes, enzymes in these vesicles will destroy anything. You probably remember the case of phagocytes, where they have lysosomes to destroy parts of microorganisms. 13. Free ribosomes, polyribosomes, so all these are used to translate RNA into proteins. Microfilaments, therefore the structure of the cell. So these filaments, they're a bit like, almost like metal cables that give structure to the cell. They're not metal, but just a bit like what a building would have to give support. 16. Microtubules, they help move things around. They're made of little tiny proteins which move along and move things around. They intervene in, for example, cell division. We saw that in the beginning of the year. Lipid droplets, storage for lipids. And that's all for the animal cells, as the cell will, chloroplasts, etc., they will not be present in animal cells. That's for plant cells. So here we are, that's the labels for animal cells. And I just forgot centrioles. They're this element here, and they also intervene in cell division to move around different parts of the cell. 1.4, ultrastructure of a mitochondrion. We've already done this, so we're not going to redo it. I will let you have a look at one of the previous videos to find the labels for the mitochondrion. 1.5, location of glycolysis, Krebs cycle, fermentation processes. There's two main things. You need to know that these processes will produce energy under the form of ATP, and it's always a question of breaking large molecules into smaller ones. When you're going to break the bonds of these bigger molecules, they will release energy, and this energy will be used to regenerate ATP. So this is a general overlook of the different things. We'll study them in the detail. So first key thing is that all starts with glucose. If you have no glucose, these reactions, they're not possible. So first thing, glucose enters the cell. Then it goes through something called glycolysis. This glycolysis is going to, through different enzymes, split glucose into two pyruvates. These pyruvates, they can either go in a process called fermentation, or they can go into the mitochondrion, this organelle here, go through different things. So TCA cycle can also be called Krebs cycle. And this process, pyruvate going through this cycle, will produce lots of ATP. You can also see that glycolysis produces some ATP. We can already say it that respiration will be more efficient in terms of energy production, because with one molecule of glucose you're going to produce much more ATP. Whereas when you go through fermentation, so when there's no dioxygen, the only ATP produced will be the one produced by glycolysis. I'm not going to go into detail quite yet, but you can see that there's NAD+, NADH. That might ring a bell. It looks a bit like NADPH. So similar function. These are molecules which will be used to transfer electrons and protons. That's what we can say for the moment. So I would advise you to learn this diagram, just have a brief idea of what's happening before we go into more complexity afterwards. Okay, well, goodbye for today, and I'll leave you with a little song to finish.